Okay, class, today we're going to start lecture 1 of Unit 7. And for this lecture, we're going to be discussing specifically the vitamins that are involved in metabolism, meaning the carbohydrate, protein, and fatty acid metabolism that we've talked about all semester long. So now we're going to talk about how these various vitamins are involved as cofactors, and hopefully the plan is that you will be able to look at the function and be able to say, oh yes, that is why we have those particular deficiency symptoms. So here we have our summary sheet, which we've seen before when we talked generically about vitamins and minerals in metabolism in Unit 6. So just showing you this again of where all the critical vitamins are, and you can see the majority of them are B vitamins, and where those are involved in the various metabolic pathways. So you can see, for instance, that biotin and niacin are important for glycolysis and various other vitamins and where they're all involved. So you can see all of these vitamins, they are all acting as cofactors, so they are binding the enzymes, causing a conformational change, allowing these reactions to occur. So without them, it would require much more energy for those reactions to occur naturally. The first vitamin we're going to talk about today is thiamine. Thiamine is also referred to as B1, and it is a pretty simple compound of pyrimidine and thiazole ring. So here you could see this upper part we have. This is the structure of thiamine. And thiamine is interesting. Some literature has an E, some does not have an E. So all throughout this lecture, I have the E associated with thiamine. And so we absorb thiamine as in the intestine, and this is the vitamin. However, within the cells, it exists as thiamine pyrophosphate. So here you could see the thiamine pyrophosphate, or TPP. So you have the pyrimidine component here, the thiazole component, and then we have this phosphate moiety, and there are so that phosphate moiety, that's where we have our pyrophosphate. And this is increasing the potential energy of the reactions. If we had thiamine in its free form with the pyrimidine and the thiazole, this is a very unstable structure. So when we have it in the diet, we usually have it conjugated to hydrochloride or a mononitrate salt. When it's oxidized, so if you have peroxidation that's occurring and reactive oxygen species, when we oxidize thiamine, it becomes thiochrome. And this is an inactive form of thiamine. So the thiochrome is unavailable to us and has no thiamine activity. So the functions of thiamine, as I mentioned, it's a cofactor, specifically a coenzyme, if you remember the difference between our cofactors and coenzymes. So it's a coenzyme that it has to bind the enzyme, cause a conformational change, and it's involved specifically more in the carbohydrate metabolism. So some of the key places that thiamine is involved is with our pyruvate dehydrogenase. So we need pyruvate dehydrogenase to convert pyruvate to acetyl-CoA. So if you remember, this is a critical step to enter the TCA cycle. So if we do not have thiamine, we're not going to be able to convert our pyruvate to acetyl-CoA. Therefore, we're going to end up having an increase in lactic acid because we're only going to have glycolysis occurring if we're deficient in thiamine. In addition, you can see that we need it for the alpha-ketoglutarate dehydrogenase, and this alpha-ketoglutarate and succinyl-CoA, this is also part of the TCA cycle, if you remember those intermediate steps. So we have acetyl-CoA is converted to citrate, and then eventually alpha-ketoglutarate and succinyl-CoA. So alpha-ketoglutarate dehydrogenase needed to convert alpha-ketoglutarate to succinyl-CoA. So this is part of the TCA cycle also. And it's involved in the pentose phosphate pathway, so it is a cofactor for transketolase that is needed. If you like to consume alcohol, we need thiamine. So we're utilizing our pyruvate dehydrogenase to break down ethanol. So we have our acetaldehyde to acetyl-CoA, and that's what pyruvate dehydrogenase is involved in. So because of this, you have a lot of individuals, alcoholics are very prone to thiamine deficiencies, which is interesting. So in addition to thiamine's function in carbohydrate metabolism, we have a couple other things that it does outside of that, including it's required for the proper degradation of branched chain amino acids. So it is a cofactor for alpha-ketoisovalerate dehydrogenase. So all of our branched chain amino acids, if we need to break them down for gluconeogenesis or ketone body formation or other functions that they have, we need thiamine in the TPP to act as a cofactor for these branched chain amino acids. In addition, it's necessary for nerve transmission. So one of the common symptoms that you end up seeing in animals that are deficient in thiamine is they have this neurological issue where they will do what we call stargazing, where they'll just put their head back and just stare up at the sky. So that is involved, that happens because of thiamine's involvement in nerve transmission. It plays a critical role in fatty acid synthesis, so we need it for the formation of acetyl-CoA. In addition, we need it for the synthesis of acetylcholine. And if you remember, acetylcholine is a neurotransmitter for the parasympathetic. And here I have a couple of the reactions that it's a cofactor for. So we have it's a cofactor for pyruvate decarboxylase and for acetylactate synthase. So basically with these, you could see that it's thiamine and thiamine pyrophosphate, or TPP. It catalyzes the cleavage of the bonds to the carbonyl carbons. So here we have our carbonyl carbon on these molecules, and we are going to be adding a hydrogen, adding lactate and onto these molecules. So this is where thiamine is involved. So it catalyzes the cleavage of these bonds, so then you can add on other components onto the molecule. So this is just a summary diagram showing where thiamine is involved in metabolism of our carbohydrates. So we need our pyruvate dehydrogenase plus thiamine to convert pyruvate to acetyl-CoA. So if we don't have thiamine, we're not going to have the formation of our acetyl-CoA to go into the TCA cycle, which requires ends up resulting in our pyruvate going to lactate. In order to bring our pyruvate to lactate, we need lactate dehydrogenase, which thiamine is also required for. So basically, you're going to end up having an abundance of pyruvate within the cell. So this is one of the reasons why with our B vitamin deficiencies, because of all these intricate pathways that it's involved in, why you get this very ambiguous deficiency symptom of just being very lethargic and having no energy, because you are unable to make proper amounts of ATP and are lacking energy. If you do happen to have enough thiamine to get you to convert pyruvate to acetyl-CoA, we need it to convert our alpha-ketoglutarate to succinyl-CoA here. So it's needed for the alpha-ketoglutarate dehydrogenase. So if it happens to get in here, this is the point in time in which it's going to stop in the Krebs cycle. So if you have some gluconeogenesis occurring, and some of those amino acids can be shunted in their metabolites, they end up forming alpha-ketoglutarate, and you think, oh, great, we could take those and continue the TCA cycle or the Krebs cycle. Well, actually, if they make alpha-ketoglutarate, then no, we're not going to be able to continue the Krebs cycle to get more ATP. So how do we absorb thiamine? So first we need to remove those phosphates. So we have our phosphatases, and our pyrophosphatases are cleaved out those phosphate groups off of the thiamine molecule, and that occurs in the upper small intestine. So more in the duodenum that this is occurring. We have both active and passive transport that occurs. So if we have low to adequate levels, we're going to have active transport. We're going to upregulate some transporters to make sure that we get increase and are able to maintain the amount of thiamine that we need. However, if you take your multivitamin every day and have excess levels of thiamine, we're going to have more passive absorption that occurs. So high concentration is going to result in passive transport. It's absorbed primarily from the jejunum in most species. Our ruminants are a little bit different. Remember that our microbes can synthesize thiamine within the rumen, and so our ruminants are actually absorbing free thiamine from the rumen. So they are absorbing it in the rumen. In addition, our hindgut fermenters, such as our horses, they have adapted and evolved to where they can actually absorb thiamine in the cecum, where they are able to get the benefits of what the microbes are able to synthesize. So here's a summary of thiamine absorption. So you can see we have our thiamine right here. If we have it in the form of thiamine monophosphate, so that's what MP stands for, is monophosphate. So we have about 0.2% can directly enter into of our thiamine, enters as thiamine monophosphate. So we cleave off one of the phosphate molecules, so we get monophosphate. About 15% of that thiamine can can passively diffuse in, and then we have 84% is what is coming in as active transport. So we are exchanging the majority of our thiamine for hydrogens. So this is where the active component comes in, because now we have to maintain this hydrogen gradient. And you see over here, we have the use of energy of our ATP in order to maintain this. We have our sodium proton pump, sodium hydrogen pump over here, transport, and we need our sodium potassium pump to accommodate. So this is where the energy is coming from. Once we have our thiamine enter, we rephosphorylate it to TPP, and that occurs by thiamine kinase. And then, as I mentioned, we have that TMP that enters, and we have some of that formed again within the enterocyte. So about 10% is able to, of TMP enters into the blood, and that occurs all passively. So it just passively leaves the cell and enters the blood. So we convert it back to thiamine, and our thiamine is secreted using the sodium potassium pump. So this is where about 80% here of our thiamine is entering into our blood. 10% of it is able to passively diffuse. Once thiamine enters the blood, it actually binds a carrier protein, and surprise, surprise, we're very creative, and this is referred to as thiamine binding protein, or TBP. It's believed to be hormonally regulated, and about 90% of it is found in the blood erythrocytes. Our thiamine can accumulate in all the cells. However, we really don't end up with significant amount of storage. The one exception to that rule is the pig. So the pig can actually store pretty significant amounts of thiamine within its tissues, but the majority of the rest of the animals, they can store some, but it's not significant, and it's not going to be prolonged like some of the fat-soluble vitamins in the liver. So we do have deficiencies within thiamine, and the majority of these incidences we end up finding because there's some sort of natural antagonism. So these antagonists either break down or bind thiamine. So we do have thiaminases, which are breaking down thiamine, and this you're going to find in your raw fish and shellfish. So if you eat a lot of sushi, you may be prone to some thiamine deficiency. They're also high in ferns and betel nuts. We have anti-thiamine substances. This includes tannic acid and caffeinic acid. And then our coccidiostat. So if you work with livestock, specifically poultry and swine, we're always battling coccidia. It is a parasite that plagues the industry, and it infects the GI tract and causes necrotic enteritis and bloody diarrhea. It's a bad thing to have. So we have these coccidiostats that kill coccidia. So one of the main commercial ones is Enprolium, and the way that this coccidiostat works is it actually blocks the thiamine transporter for the coccidia. Coccidia's scientific name is Emeria, so this is the same as coccidia. Just the fancy scientific word. So the amprolium is blocking the thiamine transporter, and that is slowing down the metabolism of that parasite and eventually will kill it. So the most common deficiency that you're going to observe with thiamine deficiency in humans is a disease that's referred to as beriberi. And this is associated with polished rice. So rice in itself in an unpolished state has adequate amounts of thiamine. However, once you polish it, make it polished white rice, those those polishings is where all the B vitamins are. So therefore, these individuals end up eating inadequate amounts of thiamine. And beriberi can be shown in two different forms. So we have wet beriberi, and this results in edema, heart failure, and skin cyanosis. And this usually occurs in individuals that have high carbohydrate diets. So if you're eating a lot of carbohydrates, you're going to get wet beriberi. Whereas if you have dry beriberi, you're going to have wasting, weak muscles within your legs and your appendages. And usually this is if you're just generally malnourished, as opposed to having the high carbohydrate diet with no thiamine. So dry is going to be just an overall malnourishment, inadequate calories in general, and low thiamine. And infants are very susceptible to thiamine deficiency, and you will see them get beriberi if their mothers are deficient, and these babies end up having heart failure, convulsions, and eventually go into a coma and die. As I mentioned before, that alcoholics are susceptible to thiamine deficiency. And so this is actually, if you get thiamine deficiency due to alcoholism, it's referred to as Wernicke-Korsakoff syndrome. So this is excessive alcohol consumption. And this occurs not only just because alcoholics typically have poor eating habits because they're more concerned about meeting their addiction of alcohol, but alcohol actually impedes the absorption of thiamine from the intestines. So, and it's basically this big vicious cycle. And as you remember, we need thiamine in order to break down ethanol or alcohol. And so if you have excess, you constantly drinking, you're going to end up with a potential thiamine deficiency. And you end up usually seeing the central nervous system dysfunction. So a lot of the stereotypical thinking of a drunk individual staggering, slurring words, etc., it's not necessarily just due to the high alcohol consumption. They could actually be showing some of those nervous system symptoms may actually be due to their thiamine deficiency, not due to the amount of alcohol that they've consumed. So here we have a picture of an individual that had wet beriberi on the left-hand side. So here we have wet beriberi, and it's kind of hard to tell from this picture, but this individual actually had pitting edema to where the physician actually pushed into the skin, and that resulted in pits. So basically, when the physician touched the skin, it did not bounce back. So if you think of if you were to touch your arm and you touch the skin right away, it bounces back and goes to the normal position that it was. Well, if you were deficient, had wet beriberi, so high carbohydrate intake with inadequate thiamine, you're going to see that you're going to leave an indentation. So that's where we have the pitting edema. And so the edema is actually where you have fluid building up around into the extracellular space. So the water within the cytosol of the cell is actually leaving, going into the extracellular space. So that's why we have the edema, and it results in pitting. Now, in the rest of the animals with thiamine deficiency, we get other symptoms. So you can get thiamine deficiency in young ruminants. So these are really young ruminants. They haven't had their microflora established. The microflora is not making adequate amounts of thiamine for them. So they are more susceptible to thiamine deficiency. In addition, if we have some of these ruminants, if they are consuming a diet that has high levels of sulfates, sulfate in particular, so it's the reduced form of sulfate, actually is an antagonist to thiamine. So it will actually bind thiamine and keep it from being absorbed by the animal. So usually in the young ruminants, what you're going to find is maybe they are consuming they're still nursing from the mother, but maybe the diet of the mother has high levels of sulfate, so she is actually deficient, and therefore her milk is deficient. So the calf doesn't have adequate thiamine within the milk. They're not going to be able to, and they don't have the microbes synthesizing it. That's where you're going to have the young ruminants have issues. When you do encounter it, because we need it for proper synthesis of acetylcholine and some other proper neurological functions, it will actually cause lesions in the brain, and which results in this bending of the neck. And that bending of the neck in all the animals is referred to as stargazing. So you'll just see the animal standing there with its head back, look like it's admiring the stars at night. So this results in a central nervous system dysfunction. In addition, you'll have hypertrophy of the heart. So we have cardiac hypertrophy to where the heart enlarges. So this is a fancy word for enlarged heart. And we see this a lot in dogs and pigs that are deficient in thiamine. So they're more prone to having an enlarged heart. So here we have a ewe that is deficient in thiamine. You can see that she's just lying there with her head up. It looks like she's admiring the stars. So that is actually, so she has stargazing. Similarly, with this chick here has stargazing. Here we have this hawk that is thiamine deficient, and it actually has neurological issues, has a difficult time flying. And here we have the heart. So this one on the right-hand side, so this is deficient. And the one on the left-hand side is normal. So you could see the hypertrophy, so this heart just gets enlarged. Next, we're going to talk about riboflavin. Riboflavin is also referred to as B2. Now, I'm not going to ask you or expect you to know the B number associated with it, but that's just more for your information if you want to see or want to know what your multivitamin, because they will list it as B2. So riboflavin functions in that it is a cofactor that accepts and transfers hydrogens from substrates. So we need it for the generation of ATP and amino acid oxidases. Riboflavin is required by more than 100 enzymes, so it's a very important cofactor, and we've actually throughout the semester been talking about it a fair amount because the cofactors, the main cofactors that it forms is this flavin mononucleotide, FMN, which we haven't really talked about, but the one we have talked about is this flavin adenine dinucleotide, or FAD. So that should ring a bell because we've discussed it a few times. So riboflavin, very critical. We've talked about all the places in which it is made throughout glycolysis and the TCA cycle, and how it actually is transferring and donating those hydrogens as it's going through the electron transport chain, which is needed to have the proper hydrogen gradient to form our ATP. So here we have the structure. The overall structure is actually FAD. So you can see the component here of our riboflavin on the left-hand side. So it's the isoalloazine and the D-ribitol are the components that are making up our riboflavin. And then we add on one phosphate group, and that is what we are making. It then becomes FMN. And then when we add a adenine onto it, so adenine is one of our nucleotides. So this is a critical component or integral component of our FAD molecule. So this is what FAD is looking becomes. You can see it's a pretty large molecule, and it's significant in many of our enzymatic reactions that we need to synthesize ATP. So when we go to absorb our riboflavin, it's similar to thiamine in that it requires active transport, and we're going to have it is absorbed in the proximal jejunum and some in the duodenum and ileum, but the primary component is going to be absorbed in the proximal jejunum. And just like with thiamine, when we have excess amounts, so if you're taking your B complex vitamins, all that excess is going to be absorbed passively, so it can be absorbed just as thiamine does. And we have non-specific hydrolases that end up dephosphorylating it at the brush border membrane. Some of it is able to bind the bile salts and is able to enter via the enterohepatic system, so it's able to be reabsorbed with the bile salts. And then some of it is rephosphorylated when it enters the blood from the enterocyte and then is transported in its phosphorylated form. So as I mentioned in the blood lecture on albumin, which is the primary protein found within the plasma, so our albumin actually is utilized to transport our riboflavin. So our riboflavin binds to the albumin, and that's how it's transported. Now, we're typically, when we're talking about these water soluble vitamins, we always think, well, it's okay to have excess because if I happen to overconsume, I don't store it, so it's just going to be excreted. But riboflavin is so critical that the liver can actually store about 33% of your various flavins. However, this 33% that's able to be stored really results in very little net storage, and that's because we are requiring so much riboflavin in order to be involved in all of these reactions. So typically, this liver storage results in very little net storage because we use it so rapidly. And we excrete it in our urine, so usually we excrete about 300 micrograms per day. So this is the amounts for humans on average. Now, if you're taking a B complex vitamin, that's probably going to be a little higher for you than those that do not. Now, deficiencies in riboflavin are relatively common. However, as I mentioned earlier when we started talking about vitamins and minerals, so in Unit 6, the majority of these deficiencies are occurring in an overall malnutrition. So naturally occurring deficiencies with specifically just riboflavin is actually pretty rare. If you have a general malnutrition situation where you are eating inadequate amounts of multiple vitamins, carbohydrates, proteins, and lipids, then you're more prone to see these. When we've had induced deficiencies, you see chelinosis, which is here on this child over here on the left-hand side, which that is basically dry, scaly lips. So you have a lot of sores on your lips. You also get seborrheic dermatitis, and this is a dry, scaly, red, itchy rash, and a lot of times it is misdiagnosed and thought to be eczema. So this is one of the, you can see on the right-hand side, a severe case of the seborrheic dermatitis. So very red, dry. Usually this is occurring primarily on the face and the scalp, not so much on the rest of your body. Now, when we have induced this deficiency in animals, we've seen various symptoms, where in humans you end up seeing this present primarily affecting the skin. You're also going to see the skin affected, but you'll see other components too. One of the primary symptoms with riboflavin, specifically in poultry, is you get this curled toe paralysis. So here we have a chicken on the upper right-hand side, and you could see that its toes are curled under. It doesn't want to stand. So we have issues associated with riboflavin, and this is mainly occurring due to its effects on the myelin sheath. So if you remember, myelin sheath is basically what is insulating all your nerves and keeping the conduction occurring. Without proper myelin sheath, you get various diseases such as Parkinson's disease. In these particular animals that were induced to have a riboflavin deficiency, you could see the sciatic nerve becomes enlarged. So here on the left-hand side, we have this chicken that has an enlarged sciatic nerve. And if you don't know where the sciatic nerve is, that is the primary nerve that is running from your back down your leg. So if you've ever had pain shooting around your hip area to your lower back, that is primarily caused by a pinched sciatic nerve. So here you could see this chicken has a enlarged sciatic nerve, which also, interestingly, many viral viruses can actually cause similar symptoms with the enlarged sciatic nerve. So it's one way to distinguish different diseases. You can think that you have a disease where actually you just have a riboflavin deficiency. You also get poor growth, and because it is affecting the skin, the epithelium, we get dry skin, and we also have diarrhea. The next B vitamin that we're going to discuss is niacin. So niacin actually has two different molecules that make up the active form of niacin. So we have nicotinic acid and nicotinamide are both have what we refer to as niacin equivalents. So we have niacin is probably more similar to our fat soluble vitamins in having more international unit requirement, but we have the two main forms of niacin. It's converted in the body to the cofactor, and we've talked about both of these. We have nicotinamide adenine dinucleotide, or NAD, and as we have discussed before, these are catabolic reactions to where we are breaking down glucose, breaking down fat is where we're forming our NADH. And then we have nicotinamide adenine dinucleotide phosphate. So we add the phosphate onto that molecule. We get NADPH. And this is involved in anabolic reactions where we're actually synthesizing molecules. So if we're doing fatty acid synthesis, we're going to be forming NADPH. So here we have the structure of NAD. You can see pretty big structure. That's one of the reasons why all throughout class we just refer to it as NAD. So you could see if we end up having a phosphate group down here, that's what's going to take it and convert it to NADP. And the protons, so both NAD and NADP accept or donate protons in chemical reactions. So here, currently, we have this as, if we add another hydrogen on here, we're going to get NADH if we have that taken off. So here is what our NADH looks like. So we have an additional hydrogen. And if we do not have that hydrogen, it's just going to be NAD. Or if we have the phosphate down here, it's going to be the same thing. So our niacin is primarily or predominantly found in its covalently bound forms. And if we have any, so the niacin that is consumed in its cofactor form is quickly hydrolyzed to nicotinamide within the small intestine, and that occurs by nonspecific pyrophosphatases. And then we have both our nicotinamide and our nicotinic acid are both able to be absorbed from the enterocyte. And this is active transport again. We have the sodium-dependent process similar with thiamine and riboflavin. And similarly, again, if we take our B vitamin complex, we're going to have, and we have excess, we're going to have passive transport at high concentrations. That can occur within the stomach. It's absorbed in the upper small intestine, so primarily in the duodenum and the proximal jejunum. and we transport it in the blood as nicotinic acid, so it's unbound. So remember, out of all these so far, this one is unbound compared to the other two. And we also have it as nicotinamide mononucleotide, also known as NMN. Now, niacin can actually be synthesized from tryptophan. If you remember, we talked about that earlier in the last unit. So it's one of the vitamins that can actually be synthesized in most animals. The rate at which it can be synthesized varies from species to species. So, therefore, for instance, 60 tryptophans are needed to make one niacin molecule in the human. In contrast, if you look at the chicken, we need a 45 to 1, so more tryptophan can be utilized to make niacin in the chicken than it can be in the human. Now, if we are going to utilize tryptophan as our source of niacin, we have to be cognizant that there are other vitamins that we need. So if we have adequate tryptophan, but just in general we have a poor diet and are deficient in B6 and riboflavin, then we are going to result in a niacin deficiency because we're not going to be able to convert our tryptophan to niacin. So we need, remember this specifically, our vitamin B6 and our riboflavin are required. You have to have them for the metabolism of our tryptophan to niacin. So to make niacin from tryptophan, we need B6 and riboflavin. And those silly obligate carnivores, such as our cats, they cannot convert tryptophan to niacin. So it's just not going to happen. But if you think about it, they never needed to adapt and evolve because they are the highest on the food chain, obligate carnivores. The diets that they were consuming had adequate amounts of niacin within all the tissues that they consumed. So they've never had to be able to convert tryptophan to niacin. So that also makes you be cognizant of, depending on the type of food, if you are feeding a plant-based diet to your cat, you want to make sure that it has specifically niacin, because your cat will not be able to convert any of that tryptophan to niacin. Now, one of the main deficiency symptoms of niacin is pellagra. And here you can see on the right-hand side, this is a picture of pellagra here. It almost looks like this individual is wearing gloves, but they are not. That is their skin. And pellagra is actually Italian, and it means rough skin. So it's a severe dermatitis. You get fissured scabs. In addition, you have a lot of mental depression. And there is some photosensitive pellagra that occurs first before you get this very severe rough skin. So that photosensitive, it almost looks like sunburn. So if you happen to be out in the sun and you're deficient in niacin, you're going to get some sort of sunburn. But the problem, or where you know that you have pellagra or niacin deficiency, is that burn or rash never goes away unless you get supplemental niacin. Pellagra was first described in the 1700s. So this occurred in the 1700s, and this happened when we had Europeans come over to the Americas. We found or were shown this great product or crop called maize or corn, and they took that corn back to Europe, and those individuals that were consuming that corn product ended up getting this pellagra, these symptoms. And for the longest time, they thought it was a disease that was transferred because all families would all get it with the consumption of the corn, but they didn't know, so they just thought it was a disease, and it really wasn't until the 1900s when it was proven that not a disease, where one brave physician actually um, took blood from an individual that had pellagra and rubbed it all over himself and proved that he would not get the skin condition. So interestingly, we also, at the same time, really did not have a lot of pellagra that occurred in Native Americans. Um, and that's because the way they utilize the corn. So corn actually does have a lot of niacin within it, but they actually utilized lime in the way they made their tortillas. They used lime and helped the corn germinate and treated it. So that process in which they utilized made the niacin more bioavailable. So that's why Native Americans that had predominantly corn diets or high corn diets, they never were deficient in niacin. Now, in the 1900s, in the USA, there was actually a significant amount of individuals, especially in lower socioeconomic classes in the South and Southeast, um, where they were plagued with pellagra. So here's a summary as to why Native Americans did not get pellagra. So they basically ended up, they boiled their corn in lime or hot alkali solution, and that actually hydrolyzed the niacin esters and made it more available. So that's why they never got pellagra. Hopi Native Americans, they harvested corn when it was still immature, so the niacin was not bound into these esters, and so it was still rather available. And at the same time, the majority of these Native Americans, they also consumed a large amount of meat and milk, and so they were able to convert the tryptophan. So if they did have inadequate or less available um, niacin within their corn, they were eating adequate amounts of tryptophan from the animal products. So that's another reason why they did not get pellagra. Now, in dogs, you actually see what we refer to as black tongue disease. This you'll see it results in ropey saliva, foul breath. The tongue gets discolored. If the dogs are fed commercially available dog food, you'll never see it. But currently, there are a lot of internet bloggers that think they know adequate amounts about dog nutrition, and they just simply don't, and you get very obscure, weird things. So this picture on the left-hand side, you can see this dog with the ropey saliva, and its tongue is, it's hard to tell, but it's actually discolored. This was actually an induced deficiency, but all of you aspiring vets out there, don't completely discount that you'll never see this, because we currently have proper diets formulated for our dogs and cats, because people will feed the weirdest things based off of what they read on a blog from an uneducated person. In poultry, you'll actually see an enlarged tibiotarsal joint. So here we have our tibiotarsal joints. So those will be enlarged. you get bowing of the legs and poor feathering and dermatitis on the heads and feet. So this is the chicken's version of pellagra, is what you'll encounter with poultry. Now, you may have a family member that has high blood cholesterol levels or high LDLs, low HDLs, and their physician may actually recommend them to take supplemental niacin. So for whatever reason, niacin is known to decrease quote unquote bad cholesterol or LDL lipoproteins. The mechanism is just not understood, but you can see here we get a decrease in our VLDLs, and because we decrease our VLDLs, we're decreasing our LDLs. So then we're going to have a reduced potential of having the oxidized LDLs if we have fewer amounts of them circulating. And at the same time, the even better thing is that we have increased HDLs, or this good cholesterol. So this is, if you remember from our fats lecture, so our HDL is synthesized from the peripheral cells, and it is carrying cholesterol, excess cholesterol, and triglycerides from the tissues to go to the liver, and then the liver is able to cause that to go in, that cholesterol gets incorporated into the bile, and we're able to excrete it in the feces. You can get some side effects if you take too much niacin. So these side effects include flushing of the skin and hyperuricemia, and all of this basically stops as soon as you stop taking your niacin supplements. Now, compared to the other B vitamins that we've talked about so far, niacin actually is toxic. So we do have an upper limit of niacin. The LD50, which is where we have 50% of the animals die at that particular dose. So, and in mice, that level is 4.5 grams per kilogram. So at that high dosage, you are going to have 50% of the individuals die. And just as we have different bioavailabilities or efficacious compounds with other vitamins, we have the same thing occur with niacin. So our nicotinamide is more bioavailable. So consequently, since this is greater bioavailable, it is more toxic. So it's 2 to 3 times more toxic than nicotinic acid. Toxicity includes vasodilation, nausea, vomiting, headaches, elevated blood sugar and uric acid, and then we can also have some liver damage. And as I said before, just like with the nutraceutical levels, as soon as you cease taking excess niacin, all these symptoms go away. You just want to stop it before you have too much liver damage, but on the upside, the liver can regenerate itself. So that amount of liver damage won't kill you unless you go crazy. Okay, the final B vitamin we're going to talk about today is biotin. So here we have the structure of biotin, and as goes with all the structures, you by no means have to memorize them. It just helps me specifically, so I assume you, be able to see what the structure looks like, how big it is, and visualize how this is working as a cofactor. So our biotin is actually bound to protein, and it usually is bound to the lysine. So it's lysyl biotin, which is lysine and biotin conjugated together. And so when we have this lysyl biotin come into the intestine, we have it needs to be cleaved, just like all the other compounds, vitamins that we need it into its smaller unit. So we actually have biotinases that are in the uh, gut mucosa that's able to free the biotin. Now, for our infants that don't have well-developed GI tracts that may not be making adequate amount of biotinases, they have free biotin is actually found within the mother's milk. We absorb it from the jejunum and ileum, and this is facilitated transport. So this again, a lot of this information seems like a broken record. So low levels to adequate levels, we're going to have facilitated transport or active transport. If we have higher levels than what we need, that's going to result in passive transport. And the microflora are able to synthesize biotin, and so our hindgut fermenters are actually able to absorb the microbial biotin, so they don't need to practice coprophagy to meet their biotin requirements. And our biotin, it does bind the albumin within the blood, but only about 12% of it is bound to the albumin. The majority of our biotin is in its free compound. So the functions of biotin include its involvement in carbon dioxide fixation or decarboxylation. And we talked about this a little bit as I mentioned when we were talking about fatty acid synthesis, that biotin is a cofactor in that first critical step. But in addition to its responsibilities as a cofactor for these decarboxylation reactions, it's also involved in gene expression. So we have some 2,000 human genes arranged in clusters, and those actually may require biotin for proper expression. In addition, it's needed for normal cell life cycle progression. So if we don't have adequate biotin, we're not going to be able to regenerate our cells. So how biotin acts as a cofactor, and we saw this a little bit when we were talking about fatty acid synthesis with acetyl-CoA carboxylase that requires biotin to convert acetyl-CoA to malonyl-CoA, which is a rate-limiting step in fatty acid synthesis. So basically, we have this carboxyl group here, and during the carboxylation reactions, you have CO2 molecules are donated by this carboxyl group that I just circled, and that is what is transferred from one active site to the other, and that's changing the conformation of the carboxylase enzyme, allowing the reaction to occur. So that's how biotin is acting in its various greater than 100 enzymes that it is a cofactor for. So biotin deficiency is pretty rare. Like I said, if you go with all the B vitamins, it's more likely to occur in a general overall malnutrition situation. You do have some inborn errors in metabolism that result in biotin deficiency in humans. In poultry, we see some biotin deficiency, especially if the poultry are fed primarily wheat based diets as opposed to corn. So wheat, although it does have biotin, is not as bioavailable for the chickens, and the microbes are able to synthesize it. So usually we don't see it very much in ruminants, but in intensive feeding situations, we can get biotin. In our hindgut fermenters, such as our horses and our pigs, they're able to absorb it within their distal intestine. So they're able to obtain the amount of biotin from what the microbes synthesize. The problem with the chickens, if you recall, that the intestine is so short that they really don't have as much time to ferment the products to meet and make the biotin more available to meet their requirements. So that's why chickens are more susceptible out of all the animals, or poultry in particular, more susceptible than a lot of the other animals we've discussed. So biotin deficiency in humans does not occur normally in adults. Usually you're going to have it, if you do see it in individuals, they're going to be adults consuming a large amount of egg whites. So there is this protein in egg whites called avidin, and this actually binds biotin. And the reason why the hen makes avidin and puts it in the egg is because that way the egg actually has 30,000 pores, and that's one of the ways in which we're limiting bacterial growth, is that if we don't have biotin available, then the bacteria are unable to have biotin as a cofactor for all their metabolic needs. And as the embryo needs it, the embryo will actually make enzymes that are going to degrade avidin, making biotin available for the embryo. So that is why they make avidin. So if you consume a large amount of egg whites, and this is mainly in raw eggs, so if you know someone that takes some chances against the bodybuilders of old used to drink raw eggs because they thought it was good for them, not concerned about the potential salmonella food poisoning that they could get. So that's typically where you saw that. In addition, you can see it more in infants when they don't have sufficient amount of biotin, either in their formula, or the mother may be slightly moderately deficient, or is putting inadequate amounts of biotin in the milk, so then the baby can become deficient. And we've also discovered a lot of inborn errors, genetic diseases that affect biotin requiring enzymes, which results in a functional biotin deficiency. And once those are discovered that an individual has those, then usually those individuals have to take higher amounts of biotin. They just have a higher requirement because they're unable to absorb it efficiently. Pregnant women can be subclinically deficient, and so that a lot of times that takes care of itself because we have them. Pregnant women should be taking a prenatal vitamin to make sure they have adequate amounts of all these B vitamins that are critical to them and the fetus. And if you have a marginal biotin deficiency, there are some papers that are showing that it may be associated with SIDS, sudden infant death syndrome, in young infants. So this is between 2 and 4 months of age. This is more likely for an infant to die of SIDS. So that is usually going to be primarily coming from inadequate amount of biotin in formulas, or mother having low milk, or those infants may actually have one of these genetic diseases requiring higher amounts of biotin for them to overcome their deficiencies in making the proteins and receptors to absorb it. So here's a picture of Avidin bound to biotin, and you can see Avidin is the pink protein, and you can see it completely encapsulates the biotin. So the circles over here is our biotin molecule. You can see the alpha helices, beta pleated sheets is our Avidin, and it's completely encapsulated biotin, and that makes it to where the biotin is not available to act as a cofactor for the various enzyme reactions that it needs. So therefore, one of the reasons why we cook eggs is so we can denature the Avidin, releasing the biotin. And once we denature the Avidin, eggs are actually a very good source of biotin, but you need to cook your eggs in order to get that nutrient out of them. Now, one of the main symptoms of biotin deficiency that occurs within, especially poultry, is what we refer to as porosis. And porosis is a condition, either genetic or nutritional. For this class, we're focusing on the nutritional components that results in slipped tendon. So the Achilles tendon in poultry, which is actually on their hock joint, slips, and it basically makes it to where they're unable to walk appropriately. So that is one of the classic signs in poultry that you will observe, and you can see if you have a substantial amount of the birds with slipped tendons or porosis, then you know that you may potentially have a biotin deficiency. Now, there are differences within poultry and their requirements. So our turkeys require more biotin as a percentage of their diet than chickens, and it's not just because turkeys are bigger, so they just have greater needs. And because of that, you're more likely to see a biotin deficiency within our turkeys. You will typically observe reduced growth and feed efficiency. And as with all the B vitamins, you'll see some sort of dermatitis. So we have disturbed broken feathers and dermatitis. We have porosis of the legs, so you have that slip tendon, and you get beak and eye deformities and incrustations. So you're going to have some, and this is more along the lines of the dermatitis is affecting the eye. And so a lot of these symptoms that I just described are a little more on the rare side, and one of the more classic signs or more common is actually fatty liver kidney syndrome, which is also referred to as FLKS. And this was actually discovered in the late 50s. So we had, and that's really when poultry industry started taking off and people moved from having backyard flocks to now we had more commercial raising of poultry. And we had, and wheat, especially back then, was one of the primary cereal grains that were provided in the diets. And you didn't so much see this dermatitis and the porosis as commonly, but we did have, when the chicks hit about 3 weeks of age, the chicks would actually just die suddenly. And when you did necropsy, you could see that the liver and the kidneys were very pale and they were encrusted in fat. And this actually occurred because the acetyl-CoA carboxylase has a greater affinity or has a higher priority for biotin than some of the other components of metabolism requiring, or some of the other enzymes. So our acetyl-CoA carboxylase got priority, and a lot of these diets had high carbohydrate, low fat, so they were high energy, lower protein and fat, and so therefore we needed to take those carbohydrates and convert them to fat. So that's why you have a lot of fatty liver and fatty kidneys. So here's some of the dermatitis on a foot pad of a chicken. So you can see that the crusting and the cracking of the toes and on the claws. And here's a diagram showing the incrustations around the eyes and the beak. So it's not so much the beak itself, but it's more of the dermal tissue around the beak and around the eyes. So that's where you're going to see more of this biotin deficiency occur in poultry. And here you can see this is a chicken that has a slip tendon or porosis, and you can see over here the Achilles tendon has slipped on the hock. So this animal's unable to walk right. Usually they will end up, if you see them in the barn, if you're walking through, they'll be walking more on their hocks because they just as if you tore your Achilles, your foot wouldn't work very well. Well, it doesn't work. Their feet don't work for them. They're unable to support themselves. And then you have some hyperkeratosis, dermatitis that's occurring on the hock also. And as with all these B vitamins, it's really uncommon to see the deficiencies in adult ruminants, but in our calves without fully functioning rumens, you're going to see some deficiency size. So they're going to have paralysis in their hindquarters, and usually you're going to have this presented in addition to vitamin K deficiency, because we're relying on the microbes to make vitamin K and biotin. So the vitamin K deficiency actually exacerbates this. We do occasionally see it in adult cattle. So our dairy cows, you can see it in the hoof horn ends up being soft and crumbling. And in beef cattle, we have records of vertical cracks in the hoofs called sand cracks that develop. And in swine and in a lot of our hindgut fermenters, you're not really going to have biotin deficiency occur, and that's because we're relying primarily on those microorganisms, and the hindgut, and biotin can diffuse across the gut wall, so we can get our biotin from the microbes. Most of the field observations that we've seen with biotin deficiency is more induced. So it's we intentionally left biotin out of the feed. And so when we've done that, we see alopecia or hair loss. You get dermatitis, just like you see with the poultry, ulcerations of the skin. You also get the cracking hooves and issues with the slip tendon, so you get hind leg spasticity within the swine. But this is not common. You're really not going to see this naturally occur. These are induced deficiencies, and this all occurred back when we first discovered a lot of these vitamins, and we wanted to know what would happen if the animal had inadequate amounts. Now we're not. Rarely are we intentionally leaving biotin out of a diet. And with the majority of our B vitamins, biotin is relatively non-toxic. Humans, we have no adverse effects if we have prolonged oral or even intravenous administration, so we're able to clear our biotin if we have excess. In swine and poultry, we've seen that we can feed it 4 to 10 times the requirement, and really we haven't seen any adverse level effects due to those high amounts of biotin. So here our conclusion is there is no toxicity of biotin in the majority of animals.